Good evening, friends. In this class, we are going to discuss about the industrial production of penicillin. So, the commercial production of penicillin involves cultivation of the fungi Penicillium notatum or Penicillium nodulans or Penicillium chrysogenum in the industrial scale fermenter and the extraction of the product as well as purification and formulation. However, in the first and foremost step to start with, we have the strain isolation, purification, strain improvement, and we will have optimization procedure for the fermentation and all the refinery process during the downstream processing of the penicillin. So that we are going to discuss about. So to look at the strains, we have the genus, especially several genera. Mainly it involves the genus Penicillium, genus Cephalosporium, and Aspergillus species, and two other actinomycetes, Streptomyces and Nocardia, and even some other bacterial species like Flavobacterium and Lysobacter are capable of biosynthesizing the penicillin using specific biochemical pathways that are associated in their cytoplasm. So to start with the biosynthesis of penicillin within the microbial cells, we need three amino acids to be present. First is L-alpha aminoadipic acid, L-cysteine, and D-valine. So these three amino acids condenses each other in the presence of the enzyme called ACV synthase. It is the pathway. So these three amino acids condenses each other in the presence of ACV synthetase. The term indicates that there will be a utilization of ATP during the synthesis of ACV in the presence of the enzyme ACV synthetase. So ACV stands for aminoadipilyl L-cysteine D-valine. So these three amino acids condenses each other to form a open ringed compound called L-alpha aminoadipilyl L-cysteine D-valine, that is ACV, and this particular product is going to be a substrate for the next enzyme isopenicillin N synthase. So this enzyme catalyzes the ring closure of the beta-lactam ring structure, which is nothing but a four-membered ring structure with participating nitrogen as well as one other sulfhydryl group that bonds with the carbon atoms of the D-valine. So a ring closure reaction will be performed by the isopenicillin N synthase to form the structure called isopenicillin N. This one looks like the basic structure of the penicillin lead molecule, and this particular isopenicillin N will be acted upon by acyl transferase to form 6-amino pymalic acid. So this is the precursor form of the penicillin. So from this 6-amino pymalic acid we will have 6 other classes of penicillin to be synthesized. But the most familiar class is penicillin G. Still it is commercially synthesized by the fungus Penicillium chrysogenum and Penicillium nodulans. So this particular structure is attached to the closed benzene ring. This benzene ring is contributed by L-aminoadipic acid molecule, which is an unusual amino acid that is still found in many microorganisms, especially in Penicillium. So after the step with acyl transferase, we will have the 6-amino pymalic acid, which is consequently converted into penicillin G, where a beta-lactam ring structure is attached to a benzyl group. So this penicillin G is otherwise called benzyl penicillin. General molecular formula for penicillin is C9H11O4N2 sulfur and R group. If the R group is a benzyl group, then the penicillin is a benzyl penicillin, or otherwise called penicillin 2 or penicillin G. If this particular beta-lactam ring structure is attached to pentenyl, 2-pentenyl, then it is said to be penicillin 1 or penicillin F. If this penicillin 3 is attached to para-hydroxybenzyl penicillin or penicillin 3, then it is penicillin X or penicillin 3. If this particular penicillin lead structure is attached to R group, that is N-heptyl form of the R group, then it is penicillin 4 or penicillin K. If this beta-lactam ring structure is attached to dihydro like amyl group, then it is N-amyl penicillin in its chemical name, and other names are said to be dihydro F penicillin. Penicillin F is different from dihydro F penicillin because there the group is pentene 2-pentenyl. Here the group is N-amyl group in case of dihydro F penicillin. And there is one other group called penicillin 5, where we find the phenoxymethyl group to be the R group going to get attached with the amide bond that is protruding outside of the penicillin lead structure. To start with the production of the benzyl penicillin, otherwise called penicillin G, the commonly used microorganism is Penicillium chrysogenum, a mold. So this was first originally isolated as Penicillium notatum by Alexander Fleming, but that has actually yielded very low quantities of penicillin. So vigorous search for the improvement of this particular strain revealed the isolation of the Penicillium chrysogenum, which gave higher yield in the penicillin production. So the newer strain of Penicillium could even produce up to 180 folds higher than the actual wild type strain that Alexander Fleming has isolated. So Penicillium chrysogenum is a kind of developed mutant from Penicillium notatum. I hope people are clear with this, and this particular mutant can generate 180 folds of the penicillin compared to the actual wild type strain that is Penicillium notatum can produce. So this one was through several mutation strategies in the genetic engineering platforms. So in actual practice, penicillin is commercially produced in submerged vat culture fermentation, employing a highly purified and selected strains of the Penicillium chrysogenum, whereby the ultimate yield of the penicillin has been enhanced at least almost threefold, that is from 10 microgram per ml to 30 microgram per ml. Only the selected strains, selected mutants of Penicillium chrysogenum can yield 10 to 30 microgram per ml of the penicillin in the fermentation medium. So interestingly, these modified, researched, purified strains of penicillin do exhibit a number of marked and pronounced cultural characteristic features, such as they give high titers of penicillin, they will have improved growth in a short period, and they have some tolerance to the side chain precursors, and they have increased acyltransferase activity so as to convert the isopenicillin N into 6-aminopymalic acid and subsequently to penicillin G form. And they have certain ability to store the intercellular requirements. I mean to say, they store the penicillin for their intracellular usage, and that can be still converted into other penicillin G form whenever it is needed. In case of inoculum preparation, so Penicillium notatum, that is Fleming's strain, together with the earlier isolate, afforded exclusively low yield. So we need to go with the Penicillium chrysogenum, that is being procured from the NRRL. It's a culture collection of the laboratory. So this particular strain, that is NRRL strain of Penicillium chrysogenum, can give good yield. So how it is developed? So here comes is the strain improvement. So you have to carefully select the mutagenic mutants using the mutagenic agents that were tested timely. It includes UV radiation, X-rays, meclorethamine, that is called the MBA. So the nitrogen mustard. Obviously, these mutagenic agents helped a long way in the appropriate selection of several higher yielding mutants. So there are two strains. One is Q176, which was also developed after the mutation, and this NRL 1951 was also a mutant of this Penicillium chrysogenum that gave rise to the formation of yellow water soluble pigment known as chrysogenin, that prominently introduced a distinct yellow tint in the final product of penicillin, because it's a mutant. The particular enzyme that is being synthesized by these enzymes will also work in acceleration to produce penicillin, and the mutants also secrete certain kind of pigment called chrysogenin, which will be imported into the final product, and this is the actual identification that Penicillium chrysogenum is involved in the production of the penicillin. Conventional methods of genetic modifications can be performed, such as people go with asexual reproduction between wild type as well as one other mutant. Parasexual recombination can also happen, and sometimes even the formation of a haploid nuclei are used for the strain development, new strain development.